sta andando. Solo su YouTube facciamo, giusto?
Sì, sì, sì. Hai provato? Ci siamo?
Sta ancora caricando.
Attenzione, dove sta l'indirizzo?
Eccoci, ci dovremmo essere.
E dov'è l'indirizzo per vederlo su YouTube?
Questa è una bella domanda. Ah, ecco, guarda, vi domando.
Ah, live on YouTube. Copy
Sta
screen.
ancora caricando. Eccoci, ci dovremmo
Sì, sì, sì.
Dov'è l'indirizzo per
Vabbè,
Quanto so belliglio su YouTube. Sembra proprio belliglio.
Mamma.
vabbè, fermiamoci.
Pure Simo è su YouTube, guardala.
Vediamo fra 10 minuti.
Vabbè. Quanto so belliglio su YouTube.
Sì, sì, sì.
Sembra proprio belliglio.
Vabbè,
Quanto so belliglio su YouTube. Ok, pare che funziona.
vabbè,
Matteo, mettiti sto sfondo per piacere.
sì, sì.
Comunque la cosa più bella è che io vedevo te e sotto John Oliver, che era l'ultima cosa. E tu che dicevi quanto sono belle e sotto John Oliver. E mi insultava Trump o cose del genere.
Ok, quindi adesso
Ok.
siamo live su YouTube e poi dobbiamo registrare allo stesso tempo, giusto?
Sì. Quindi alle 2 come iniziamo metti record. Se mi metti
Metto
co-host lo posso iniziare io, penso.
Ah, c'hai ragione, fammi mettere... Dov'è Matteo? Make cost. E metto cost anche Simo, giusto in caso ti perdiamo.
Eh no, più che altro che deve cacciare la gente nel caso ci abbiamo problemi.
E Sila ha detto che fa dentro e fuori appena
Sì,
può,
sì, sì.
quindi non la faccio cost e poi faccio
Ok.
Chantal appena si connette.
E io
Magari
fra 5 minuti metto il link di YouTube su Twitter. Quindi entro i prossimi 5 minuti dobbiamo sparire perché siamo già su YouTube, altrimenti le persone ci vedono qua a conversare.
aspetta un attimo che così intanto vado a fare la pipì.
Ok. Uh, ok, quindi siamo... Tutto a posto allora.
Sì, poi quando arrivo più o meno alla parte dei pitch,
Mhm.
vabbè, in realtà lo posso fare io perché man mano tu sei il primo perché il tuo progetto, quello con Simone, è il primo su GitHub, poi man mano che andiamo sugli altri dobbiamo fare le persone panelist, tanto abbiamo tempo. Ah, Davide Mumi se n'è andato.
Eh beh. Ma quindi, vabbè, tanto è velocissimo fare le panelist. E poi dopo gli dai...
Sì,
Qui c'è già Who
l'unica
can
cosa è che
share.
se si presentano 90 persone devi praticamente scorrere 90 persone e cercarlo.
Ah, giusto, giusto. Vabbè.
Come si cacciano le persone?
Se vai su More,
Sì, però
c'è...
al more degli attendees, perché non penso che puoi cacciare i panelist. Comunque sì, a fianco ai nomi in attendees, che adesso è 0, dovresti vederli.
Ok, va bene, poi controllo.
Voi potete vedere tutti i comandi che ci sono sotto? Adesso che
Tu-
siete cost.
tutti tranne la roba di YouTube e qualcos'altro.
Ah,
Ok.
io sì, vedo record, share screen e basta, Q&A c'era anche prima. E chat.
Ok. Ok, del record, chi lo premi tu, Matteo? Il
Sì, sì, sì,
record?
prima di iniziare a parlare.
Ok.
E l'ultima domanda, ma scusa un po', ma come funziona? Noi che abbiamo organizzato qualcosa, poi però non c'entriamo niente con i progetti vari, o sì?
Cioè, nel senso, tu c'è un progetto con Marco, mi sembra,
Vabbè,
no?
ma quello ok, però io tipo non c'entro niente col tuo progetto o tipo noi organizzatori siamo come il prezzemolo e c'entriamo con tutti i progetti?
No, no, nel senso, se ci sta un progetto che ti interessa, puoi andare a parlare con le persone, però no, ufficialmente non devi fare niente.
Ok. No, era ufficialmente se esce qualcosa, noi dobbiamo, tipo metti che esca qualcosa nel mio e Marco, Vigliami, Sila e Chantal e te.
E Matteo.
No, 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 cioè le persone che hanno lavorato al progetto.
Ok. Quindi niente, noi abbiamo solo la gloria di aver organizzato.
Esatto.
Ma
Che non è poco, che non è poco. Ok, c'è un attendee. Ah, Chantal, ce l'è. Oh, Alessandro, ok. Promote panelist. Alessandro anche panelist, Matteo?
no.
Beh, no, tu lo
No.
devi fare il pitch, non lui.
Ok.
Hello.
Hi, Chantal.
Ok, posso fare allow to talk?
Can you see also how many people join or not?
Yeah, so far one, apart from us.
Ale, can you try speaking?
Now,
Okay.
Yeah.
you hear me?
Yeah, perfect.
But you don't see me, do you?
No.
No.
No, but that's normal. It's a webinar.
Yeah.
You're not supposed to
Yeah.
see me.
Yeah, but I can enable you talking or I can kick you out.
Kick me out, please.
They gave me this power.
You will regret this.
Okay, Chantal.
And can you try also the Q&A, Ale, since you are the only one attending?
Okay, I can even raise a hand. Do you see anything?
Yes.
Yeah,
Okay.
And I can lower.
Now I lower my hand.
Okay.
thank you.
Q&A.
We are also streaming on YouTube, Ale.
Okay, so I change the question.
Just in case.
I cannot seem to get this background working. I think I just have a too old laptop or something.
Oh,
That's the question.
I just look like a
Yeah,
ghost.
sorry, my keyboard is broken.
Okay, we can do this. Answer live. Done. Answer.
it goes in
No,
answers.
I don't see it.
Okay, excellent. It went into
you
answer.
don't see. I think you see if I reply. Try another one.
No, I don't see the question. This question has been answered live.
Yes.
But I don't see any answer.
No, because I answered orally.
Okay.
Try to write another one.
Okay.
So type an answer.
Ooh.
We're going to say hello
Oh.
quickly.
Hello.
Hello.
Hey, cutie.
Oh.
Hey!
I have to go get changed.
Yes. Look at that.
Okay. I'll come back in a bit.
See you soon.
Ale, do you see the answer now?
Yes.
Good.
Yeah. Okay.
And
Excellent.
now I can do delete. Open anonymous.
Anonymous question.
Haha. Guess who am I?
So I will do dismiss. Don't scare us.
But what's the difference between then the Q&A and the chat? There's also chat.
Chat?
Yeah. So in the chat, basically, people can talk freely, and well, the Q&A is just for asking questions that one would like to be answered from the panelists.
So this I can send to everyone, to the panelists.
Mhm.
Everyone and panelists.
Yes. And this hi everyone.
Yeah, exactly.
Now I don't have any more buttons to click, Simo, sorry.
Okay, now I can mute you back.
Okay, good.
Bye, Ali. It was a pleasure.
Ciao, ciao.
Alexander, hi, welcome.
Hello.
Hello.
Should I enable him as well? Allowed to talk. Ask to unmute. Then I will stop playing.
Yeah.
Hello, can you hear me?
Yeah, hi.
perfectly.
Can you also see me?
No.
Yes or no?
No.
All right. Let's see where I can adjust this.
But I think with
No.
purpose.
Aha, so you're explicitly...
Don't want to see people.
Yeah, that's a good thing.
I think we needed to ask the agreement to everyone in case we wanted to show images, so it's less painful.
All right. I see I can mute myself or not, I guess, right?
Yeah. Yeah.
All right.
Okay, so I'm about to tweet the YouTube link. So after that, in 2 minutes, we can start and I will hit record.
Okay. So should I disable everyone from the attendees?
Yeah.
Only
Looks
panelists.
more okay.
So as people are joining, hello everyone, and if you would like to invite anyone else to follow this session, even if they are not registered, they can join us on YouTube. I just put the link on the chat. And we are at 13 attendees, so let's wait just a couple of minutes so people that just remembered that something was starting now can join in and hopefully find the right link in an email that probably it's in the spam folder.
Should we share the YouTube link on Twitter as well?
Yeah, I just tweeted. So if anyone wants to retweet that, it would be great. So a little spoiler from the presentation, we actually reached, if we count the organizers, the triple digits, we should be 100 registered participants. Now, let's see how many real people will actually join the meeting.
So supposedly some people have problem with the link because when they click on the link, then it asks them to log in with a UCL account. Was this a problem with some of the attendees as well?
I don't know, for me, it asked me the password.
Yeah.
When I copy paste on Zoom directly, but when I click on the link and open from the browser in the app, no password.
To me, it asked just, it had my name and it asked my email, and I put my Gmail email, and it went through.
Okay, you have to be logged
in on Zoom, probably, but it shouldn't matter if it's...
For me, it asked the name of my first pet. All right, so... We are at 26. So since it's almost 05, should we slowly start and then people hopefully will catch up? Yeah, we are at 27 attendees. I think that we can definitely start. So I will hit record. Okay, so hi everyone. I hope that all the people that I saw in the registration forms will join soon. But in the meantime, let's slowly start. Welcome to BrainHack Micro2Macro, and I will start sharing my slides. This will be just a quick introduction to tell a bit about the format, about what we thought, and about what will come today and in the next days. So first, this is a satellite of the BrainHack Global 2020. Actually, we are now in 2021, so this is the very last satellite event of BrainHack 2020, and actually the first one of 2021. There is a website for BrainHack Global that include all the projects that have been submitted so far, including the projects submitted to this event. So if you are curious about that, go check the galleries because there is a lot of cool stuff there. And let's meet the organizers. Probably you know all of us, and I just wanted to take this chance to show you the faces that organize these, and to thank all my co-organizers. And if you want to reach us because you have any issue, please do. You will know some of us. And thank you for registering, because basically today we reached, as I spoiled before starting the recording, the 100 registered participants. And I was quite amazed at the actual distribution, because we thought this event to be kind of shared between the East Coast of the Americas and Europe and Africa, and in terms of time zone. But then we also got some unexpected registrations from China, Australia, and even Mexico and Western Canada. And some words about the format. So it's supposed to be a couple of intensive days. Of course, since it's a virtual event, it's up to you how much intensive it will be. But the idea would be that Today there will be the pitches for the projects, and you will be able to join a project that interests you, and to not just spending time writing code and testing workflows on data and things like that. There also will be unconferences, and we will see the schedule in a bit. And one thing that I want to mention is basically, one is never sure about submitting a project or not, but if it happens that you get a crazy idea at the last minute, even today, and you want to try to make this happening, just get in touch with us on Mattermost, and we will create for you a breakout room for the hacking sessions, and it will actually happen even if it's last minute. So you may have seen that in the schedule there is a slot for hacking time, but actually hacking time can be any spare time that you have in these next days. And we wanted to actually reserve some slots just to have a reference, so people, if want to actually hack together, they know that in that time they can, and it should be a time that it's feasible for both people in the US, in Canada, and in Europe. Maybe a bit less for people that register from unexpected place, but hopefully it will work out. And of course, if you are in a project and you think that you want to have a time where it's more feasible to you to all work together, please set it up. It's okay, of course. So a lot of people may be new to this kind of format, so I just wanted to kind of share what a BrainHack project could look like. So I will just briefly share my experience at the latest OHBM BrainHack. So I had a coding projects where I wanted to implement in a toolbox that I wrote some features, basically these 3 features. So I pitched my project, and I found someone else that wanted to join in. So it was the 2 of us. So basically we divided these 3 features among ourselves. So he got these 2 that were kind of smaller, so they should have been feasible. So he implemented the first one in one day, and then in one morning he implemented the second one. So we were all very happy, except that in the meantime I was working on the other feature that was kind of bigger, and I spent some time on the first day trying to fighting with Docker and making the Docker image small without succeeding it. And in the end I just set out with a big Docker image. And then I found a bug in NiPipe that was the toolbox that I needed to use. I managed to fix it talking with the developers of NiPipe. Then I found another bug. This was all in the second day. And then I fixed it also, this one, thanks to the amazing NiPipe developers. And finally, I found a path issue. This was on the morning of the final project's presentations. But then I managed to fix it in time for the time of the final presentations. So this is to say that actually things can go very bad, but it can be fun anyways. And if you actually find bugs, as happened to me, actually it's a good thing, because it's better to find them in these occasions and not in let's say times where you don't want to find an unexpected bug in your code. And if you get stuck and you have problems with the software that you didn't actually write yourself, please try to get in touch with the developers, because most of the time that they will answer, and sometimes even faster than you expect. So let's have a look at the schedule. So today, during this session, we will have the project pitches. Then we will have basically a 2 hours time slot where you can talk with the project leaders. And this will happen on the Zoom hacking space. That is the other link that you should have received in the email. And then we will have a presentation by Qmenta. And then there will be the trivia night. Wednesday instead, we will have the unconference and some hacking time, the same Thursday. And finally, on Friday, we will have the project presentations, and to wrap up, the presentation of BrainHack Proceedings, that is a new project from the BrainHack group. So I think that as amazing as the number and distribution of participants are the actual speakers of this BrainHack, for the first session that will be on brain microstructure, we basically have 4 speakers, 2 of them from Argentina, 1 from Lund University, and another one based in Portugal. So basically, we have quite a spread selection of speakers, and also we complete the picture with the second session focused this time on machine learning, that will be from people from China and people from the US. So I really feel the international atmosphere of this event. And I mentioned Qmenta. Qmenta is sponsoring this event. So basically, if you have an idea and you maybe think that you don't have the actual computational resources to run it, or if you want to actually drive your project towards reproducibility or towards ways to actually making tools available and making them easy to use for people that maybe they are not accustomed to coding, please get in touch with them, and please join the session that we will have later today, so you can ask them directly questions on how they can actually support your project. And last but not least, at the end of today, we will have the social event. I have not sent yet the link, but basically it will happen on YouTube, and it's on you if you would like to actually follow it with other people on the Zoom hacking space, or if you prefer just to take a break from Zoom and then you can just watch it on YouTube on the TV if you want, and you can just participate to the quiz with a phone. So it was thought to let you have the choice to even be a lot of engaged, or just to get a break from time at the computer. And finally, I think it's time for pitches. So we have 6 projects. Basically, we were almost having the double of the numbers that we had for most days. And I think they are all amazing, and the colors of these slides probably say it all. And I would like to start then with the first pitch, and I will see you then later in the hacking space. Let me stop sharing my screen. So the first pitch, it's from Marco Palombo. Marco.
Yes, thanks, Matteo. I will share the screen. Can you see my slides? Can you see the pointer?
Yes, I can see your mouse.
Okay, excellent. So thanks, Matteo, and welcome to this first project pitch. Our project is called the Sandy in Amico, and how you can imagine from the title, we propose to implement Sandy in the Amico framework. So briefly, what is Sandy? Sandy is just another compartmental model. What is special about it is that for the first time, we asked a simple question. Given a neural cell, which can have a shape, complex shape like this, is it possible that the signal coming from the cell body, which we call soma, and the signal coming from neurites have 2 different signatures on the diffusion MRI signal? And if so, can we measure it? Last year, we proposed Sandy as a way of measuring indeed the apparent contribution of the soma and the neurites to the signal. And Sandy itself stands for Soma Neurite Density Imaging, and it is a compartmental model where the intrasoma signal is modeled as a restriction in sphere of a given radius R. Then the neurites are modeled as randomly oriented sticks, and the extracellular space is modeled as a Gaussian isotropic diffusion. Sandy uses the direction averaged signal so that this assumption can be more likely met. And we have shown in both human and mouse brain that the contrast of sphere signal fraction remarkably matches our expectation from the BrainSat architecture. However, there's a problem that Sandy, as all the multi-compartmental model, is extremely challenging to fit, even if you have a rich data set. So one of the problem is not only that it is slow and there are local minima, but also that the fitting algorithm depends on the initial conditions. And if you use machine learning algorithms, like in the original work, the architecture and the training strategy can substantially impact your prediction. So what we would like to do is to provide a more robust and agnostic fitting pipeline for this multi-compartmental model. To do so, we propose to use the AMICO framework, which stands for Accelerated Microstructure Imaging via Convex Optimization, which in Italian also means amico, which means a friend. So we hope to find in AMICO a good friend for Sandy. And the basic idea beyond AMICO is to beat the two main problems of conventional fitting of multi-compartmental model. One is that they are very slow, and the other is that they, as I said, suffer of finding a spurious solution blocked in local minima of the objective function. So the idea is to accelerate the fit and to simplify the fitting landscape by linearizing the problem. And that's what AMICO basically does. It writes the problem in a convenient linear form and find the solution of the problem using efficient optimization. So in this example, you can see, for example, the formulation in AMICO of NODDI, where the intracellular signal is comprised of a dictionary of pre-computed signal coming from the formulation in NODDI of the intracellular signal or intraaxonal signal. Then you have the extracellular or extraaxonal space, again, a dictionary of response function for the signal given a specific model, and then the isotropic one. So basically, AMICO rewrites virtually any kind of compartmental model in this formalism and can solve it very efficiently and quickly. This was the example of the application of AMICO for NODDI, where you can see it provides very similar parametric maps, but in much faster way, 6 minutes versus 65 hours. So what we propose to do in the project is to implement different aspects of the Sandy processing analysis pipeline within AMICO. So first, you need to download from the Git repository of AMICO, the AMICO Python version, in which you will see there will be a Python code to load the data, Python code to generate the data set, one to perform the fit, one to save the data. So what we aim to do is to modify these classes and functions in the Python AMICO code to accommodate Sandy as a model. So first we need to compute the direction average signal in each B shell, and then we need to define the Sandy model and the response function to generate the data set. I don't think we need to change much about the optimizer and neither about saving the data, but we will discover this during the ITER. It would be nice to also start writing a tutorial wiki page on how to use this Sandy Amico version we will achieve. So what we would like to have at the end of the 3 days is a tested and stable release of Sandy Amico to be released on the GitHub where Amico is, and also wrap up an abstract for OHPM, whose deadline is 1st of February, to describe this new toolbox that hopefully will be ready to be shared with the community. One last comment about the dataset is a not published dataset, so you can, whoever is interested can get the dataset from me. It has been provided by Andrada Janusz and Noam Shemesh at the Champollion Center for the Unknown in Lisbon. And as you can see here, this is the logarithm of the direction average, the normalized signal. The dataset is of very good quality. It's in vivo, it's mouse brain. It goes up to 12,000 seconds per millimeter square in B-value, resolution in plane of 120 micron, and has diffusion times compatible with the Sandy model. The SNR is pretty good in both white matter and gray matter. So we hope to use this dataset as a good test set to test our pipeline. So if you are interested, please get in touch with me or Simona Schiavi via Mattermost so that we can make you join our channel and we can proceed from there. Thank you.
Thanks, Marco. So let's go along the list of the projects on GitHub. Next, we have Viliami with what to do with the shitty clinical data, how to apply your perfect estimator model method to data that contains outliers. Viliami?
Okay, thank you. You should see my screen now.
Yes.
And can you hear me?
Yes.
Yes, great. Okay, so my name is Viliami, and the title of my project is What to Do with Shaky Clinical Data. There is one extra H in mix. And if you want to see the web page of this project, there should be QR code in each slide, so you can open it with your phone. And you have my contact information on that page as well. So as a background, in modeling the brain microstructure, you usually have 3 things. You have the model, you have the data, and you have the estimator. And usually depending on the model, you consider things like how well it represents brain structure, or does it represent the diffusion weighted image signal. So for example, the diffusion tensor perhaps represents the signal better than microstructural parts of the brain. And for the data, you should consider how good quality data is. Does it contain artifacts or outliers? And thirdly, the estimator basically is the algorithm that computes the model coefficients based from the data. And it should be robust to outliers and artifacts, and it should also minimize the error propagation to the model from the measurements. This project aims to improve the microstructural modeling and diffusion-weighted signal representations in the third section of the estimators. So currently, many estimators are evaluated only with good quality data, such as Human Connectome project, and which is shown on the bottom. And there is not much of noise and no outliers. And then somebody would like to apply that new model and with its estimator to a clinical data set, shown also on the bottom. And that could go very badly wrong, because the clinical data can have motion artifacts, it can have very weird outliers, and more noise. So our aim of this project would be to generate some kind of data set to evaluate new methods, especially the estimators, and compare already existing ones, and perhaps even make some of them robust to these outliers. To motivate you, let's compare DTI versus NODI. DTI has been around since 95 about, and NODI has been about 2012. And these numbers are from Web of Science, and DTI gets still about 1,500 hits annually, whereas NODI gets nearly 100. So why is this so different? While NODI could explain so much more of the brain structure than DTI, why DTI is still used? I think it's because it's easy to apply. It's robust outliers. There's many good tools to process diffusion-weighted images and fit tensor model on it, even if the data has outliers. And this slide shows the comparison, or which kind of fields of science use diffusion tensor imaging. In total, there is over 20,000 hits on Web of Science on diffusion tensor imaging. Most of the users are neuroscientists and clinical neurology and radiology. Then there is neuroimaging and surgery as well, and psychiatry. So there is large medical fields using DTI. And if we compare this to NODI, there is clinical neurology basically, and 1% of the DTI hits. So NODI is not used. It's a neural model, but it's still not used as much as it could be used. And I would like to fix this and perhaps help other new models as well in the process. So to reach this, I have generated this realistic diffusion-weighted image simulation from Human Connector project data with MRTrix, probabilistic tractography, and microstructural modeling using COMIT. And I have added outliers and noise. So you can, switching diffusion-weighted images, you can see those stripe artifacts. It's a really basic simulation of subject motion during the scan. But this dataset should be good enough for evaluation of different microstructural models and their estimators. I would aim to investigate different toolboxes and libraries available in Python and MATLAB mostly, and see if those, or evaluate how robust those estimators already are using this dataset. And if they are not robust enough, then I would investigate how to make them robust. I don't think everyone or all algorithms can be made robust, but maybe some of them can. And one additional thing to provide here is this dataset. So in the future evaluations of new estimators and models, the same dataset could be used as a benchmarking tool. So hopefully this leads up to a paper. It depends on the results, of course. And that's pretty much my project. Happy to hear if it raises interests.
Thank you, Viljami. Can you stop sharing your screen?
Yes.
Thanks. Thanks for the presentation. And next we have Christine Koller from Kubrick. So I will make her a panelist. If I manage to find her. Yes, here it is. Hi, Christine.
Hi, everyone. I just have to share my screen. Can everybody see my presentation? Okay.
Yeah, we can see it.
So, okay, so hi everyone. I'm Kristen, for those of you who don't know me. I'm a postdoc here at Kubrick, and thanks for coming to BrainHack. For this project, the question we are continuing to ask is, when we measure the same thing twice in our MRI research, or even multiple times, do we get the same result? Or do we at least get something close enough so that we can use it in our studies? So just to give a bit of background to the project, and I should have mentioned, there's a group of people from Kubrick. We all came up with the project together. I'll list their names again at the end. So just to give a bit of background, we recently published a paper where we tested repeatability across 5 MRI scans in 6 people, 6 healthy humans, using multi-shell diffusion, multi-component relaxometry, and quantitative magnetization transfer sequences on the Connectome here at Kubrick. So in the top left, you can see we looked at repeatability in 3 different tracts. So the fornix, corticospinal tract, and orchiofasciculus. And then we mapped these tracts onto the microstructural maps on the top right. And those we derived from the sequences we collected. So we essentially looked at a tract-based repeatability where we averaged all of the values along the tract. And we also did a whole brain analysis. Picture is a bit small. I don't know if you can see. Can people see my pointer actually? You can, okay. So you can at least see that roughly individual scans were all correlated, and overall the repeatability results were really good. So we had high ICC values between, we had fairly high ICC values between 0.72 to 0.98, and very low percentages of coefficients of variation. So these are measures that people typically use to assess repeatability. But basically, the question that we are now trying to ask is there a more informative way to do this? So rather than averaging along a whole tract, is it better, or can we get more information if we look at different properties of the tract, such as the endpoint, size, or streamline number, or hopefully any other properties that you guys might have in mind? So that's one goal that we have for our project. Other things include investigating additional strategies, for example, extra steps that you might include in your processing that we might find out that there's a crucial cleanup step that really affects your results that we haven't looked at before. So then we also have a bunch of kind of branch off ideas for the project. So basically, we want to keep it fairly flexible. If there's something you want to do with the data, you can do it. But yeah, we have a couple of branch off ideas if people are interested in exploring maybe processing pipelines, or even looking at different tracts dissected using different tools. So do we get the same result visually and quantitatively if we dissect, for example, the corticospinal tract using MR-Trix compared to FSL, or whichever tool you might be interested in using? So the goal is really to challenge, not necessarily challenge existing methods or ways to measure repeatability, because a lot of them are good already. It's just trying to find more informative ways where we can get a different or more informative information of what repeatability means. So just a little bit about the data. I don't know if people have checked out the GitHub description of the project, but the data is publicly available. It's part of the micro data set that I mentioned before. So it's basically 5 MRI scans in 6 healthy humans, and it includes diffusion data, multi-shell diffusion, multi-component relaxometry, and QMT. And we have the raw data and process maps, so things like FA and D, and all of the the maps are on there, mile and water fraction, things like that. All of those are there. But for this project, we're also making available the tractography results. So we did the tractography in MR-Trix. So the TCK files are on there already, but we are looking at making additional files available if people want to have more control over what they're doing and if they don't want to process the data from scratch. So hopefully that gave a little bit of an idea of what we're planning to do. And as I said, if anybody has an idea of what they want to do, please come and join us. And we just want to have fun with it. And so everybody is welcome, no matter what your skill level. And so just, that's, maybe we can put your name on the collaborator list. And there's a link to the GitHub page there with some more resources as well. That's it for me.
Thanks, Kristin. It was a very nice project. Can you stop sharing your screen?
Yeah, I think. Sure.
Yeah, thanks. So the next one on the list is Paddy. Paddy Slater from UCL. I had it just here. Here is it. Let's make him panelist as well. Hey, Paddy.
Hi there. Hi, Matteo. Hi, everyone. Right, let's just share my screen. So I'll assume that you can see everything and hear me.
Yeah, perfect.
Okay, thanks very much. So yeah, this is my project. I'm presenting for BrainHack here. So in short, what we're looking to do is to do some microstructure model fitting in the brain, but do it a little bit differently to how it's normally done by effectively sharing information across voxels to kind of improve these microstructure model fits in the brain. So I won't spend too much time on the background, but if we assume we have some diffusion MRI data, which in a sort of Bayesian way here, I'm just going to call Y, then we can call our microstructure model some function of some parameters theta, and it gives us some microstructure maps of those parameters theta. So a microstructure model is just a way of connecting the data to some parameters. That's how we can think of it in a sort of Bayesian way. So typically this, obviously, the microstructure model is fit with a voxel by voxel least squares fit. That's like 99% of the time, that's how it will be done. Yeah, I was recently interested in this paper from a few years ago, actually, which takes a kind of different approach. And so this is essentially what they've done here is they've implemented they've thought of the microstructure modeling not as a sort of voxel by voxel process, but as a Bayesian hierarchical model, where there's sort of, as well as your voxel-wise parameters, you have like an overriding prior on those parameters, which, as you can see pretty clearly from these maps here, these are liver maps with a simple IVIM model, which is a pretty simple two-compartment model. And they can show that when they use a Bayesian hierarchical model here, rather than just simple least squares fitting, they get a massive improvement in their parameter maps. So essentially, the overall aim of this project is to take that idea from that's been applied in the liver for a pretty simple IVIM model and implement it up for some more complex brain microstructure models. So here you can see in my picture here, on the left-hand side here, we have the posterior distribution, which by Bayes' rule is proportionate to the prior distribution. times the likelihood. So typically in a normal least squares model fit, you're only really accounting for the likelihood there. But when we look at doing a Bayesian hierarchical model, we also have this kind of prior distribution over the parameters. So and that allows us to estimate this posterior distribution over our microstructure parameters on the left here. So in terms of like more concrete deliverables, so it would be really cool if we could manage to code up the implementation of this Bayesian model, which is with a which we kind of infer with a Markov chain Monte Carlo algorithm. First of all, we'll try and test that on simulations to show that we can kind of accurately infer simulated microstructure model parameter maps. And then the second big concrete goal is to take this MCMC algorithm and apply it to a single HCP diffusion data set to do this Bayesian hierarchical fitting on some in vivo data. So as sort of smaller tasks, which we would be looking to work on towards these deliverables. So for the first one, I think first of all, we have to have a discussion about which models would be best to sort of try and apply here. So that's something that we can discuss based on people's interests. Then we need to try and simulate some simple test data sets. And so for this and for much of the coding in the project, we'll be using the DMIPy Python toolbox, which hopefully a few people are familiar with. And then so given this kind of first simulation, we'll then go on and implement our MCMC algorithm, which will infer a Bayesian hierarchical version of some brain microstructure models. So for this, we'll kind of use the DMIPy software library and sort of add a bit of our own original coding to that to implement this MCMC algorithm. And then on the other side, on the kind of in vivo data set side, which is a task that can kind of go on in parallel with that first task. So for deliverable 2, we need to download choose download a suitable pre-processed HCP scan, do some segmentation on that, perhaps with SBM or FSL packages. And then we want to implement a baseline least squares fitting on that data set. And then finally, the big overall aim, final task is to apply our MCMC algorithm to that HCP scan. So in terms of the skills which would be useful for this, so by no means do you need to have all of these or be an expert in anything, but these are the kind of skills that we would be using. So as I mentioned, we're going to use DMIPy, Python toolbox, Jupyter notebooks. I think this is kind of quite a sort of a hacking away kind of project. So I don't think we'll be, certainly not in the first couple of days, we won't be like trying to integrate, smartly written code into anything like that. Anything will be sort of hacking away on Jupyter notebooks. So if anyone has anybody with familiarity with brain diffusion data processing and HCP data, that would be especially good, because I should mention that don't usually work on the brain. I'm a bit of not a brain expert by any means. So that would be that kind of expertise would be really useful. And hopefully, because this is BrainHack, there'll be plenty of people with that. And then the final two things are, which will be useful, but also, I mean, these are things that you can just learn as you go through the project. A kind of basic understanding of model fitting methods, and also Bayesian statistics as well. Kind of basic understanding of those things, and I guess also a basic understanding of Markov chain Monte Carlo algorithms. Yeah, so as I mentioned, I think the two main learning things that you can learn from this project are Bayesian hierarchical modeling and MCMC. Personally, I want to learn a bit more about these brain microstructure models and working with HCP data, because I haven't done that very much in the past, or at all really. And so, yeah, that's everything. Here's a few more details, and obviously reach out on the Zoom breakout rooms in a bit if you have any more questions, but hopefully this has given you a bit of background of this project. Thanks for listening.
Thanks, Paddy. Very interesting project as well. I remember that there are a few participants that put Bayesian approaches as their specialty, so you should be able to find those. So now, next on the GitHub list, it's me. So let's share the screen. Okay, so this was kind of last day, last minute project in the sense that I started thinking about this in the last 2-3 days, and then last night I decided to write it down and submit a project about it. And it's entitled Microstructural Variation on a DIM, a bottom-up approach to diffusion models. So why this project? I think that what has always fascinated me about diffusion MRI is the fact that when you want to study white matter microstructure, you can do a lot of stuff. And it's, in the end, always a problem of compartment models in a way. And actually, the literature is a good picture of it, because we have so many variations on what someone has called the standard model of microstructure, that basically is seeing the axons as sticks with the water that may either be inside there or outside. And the problem is that since there are so many models, it becomes hard for people that are not inside the hardcore MRI group to understand what they should use and why something would be better than something else. And I think as a whole, this is the problem with studying the brain is that since it has attracted so many fields on it, and it has become so multidisciplinary as an environment, it also has inherited kind of a collateral effect that is some fields are more pure than others, as this XKCD cartoon shows. And as a result, most of the time you will have conversations between people working on applied stuff, or even clinicians, and people from the physics side that will go a bit like this, like that there is kind of an incommunicability wall behind them. So the idea for my project is basically focus on what is kind of the thing that most people agree we can measure using diffusion. At least we can estimate, that is the intracellular volume fraction as a signal. And basically, since we have so many models that have been published that we can use to estimate that, I'm curious to see what happens when you start to compare all of them, to really understand how big is the actual difference that you will get in these parameter maps, and how small the difference may become when you move, for example, from high quality, ultra high gradients data to lower quality clinical grade data. And also what happens in pathology, to see if actually you will have consistent results across these models. So in terms of specific goals, what I thought was to start putting together some workflows for each of these models that can be easily reproduced by anyone, and then start to run these workflows on different data sets, and start with even very simple comparisons to begin with, and then think about maybe if things seem interesting to other people, to how to follow up this, because since it's we still need to figure out how actually this project will look like in the end. It's probably we will need time after the hackathon, and also take into account that we have Qmenta that can support this kind of initiatives. In terms of skills and data, actually on the skill side, of course, if someone is experienced with diffusion MRI and microstructural toolbox, microstructural toolboxes is better, but it's not that strict as a rule. Actually, the
the most desirable skills that I didn't write here is that you would like to learn to use one of these tools. So if you want to learn how to use MDT, you will do it in this next 2 days, and we will end up at the end with a script that can do charmed using MDT or something like that. And in terms of data sets, as Kristen already mentioned, the micro data sets, that one would be an interesting one to use, since it has been acquired on the Connectome scanner, and it has also scan rescan data if we want to look at that. And then start to dig around in OpenNeuro, DataLab to have a sense of which kind of realistic data set we can use. And when I say realistic data set, I'm referring to data set where you don't have a lot of b-values, you don't have a lot of directions, like data sets that you may end up with and you wouldn't say, this is a beautiful data set. And in terms of good first issues, it's quite a list, because basically, since the starting point is actually having these workflows implemented and easy to be run, a lot of first issues would be just to actually implement of each of these. So for example, implementing node-based workflows using a Micro, or using MDT to estimate charmed, and so on. And other good first issues that are not, let's say, coding related would be to identify the good data sets to use. I always spend a lot of time on OpenNeuro, because there aren't so many diffusion data sets, and it would be good to have an idea of which ones will work for this kind of project. And then another thing that would be good to figure out is what would be the most efficient way to actually compare all of these approaches, because of course, as a starting point, we can just start looking at the correlations between pairs of them, but as the number of workflows increase, and as we want more quantitative data, it wouldn't be the ideal. So that's that as well. And actually, that's it. I think that mine was kind of on the short side, but it's okay. So we saved time. And if you're curious about the project, get in touch. Either start writing on the Mattermost channel, microstructural variations, or come later to the breakout room in the Zoom hacking space. Thank you. And now, I have the last one that should be Conrad, if I can find it. Conrad, can you hear us?
Can you hear me?
Yeah.
Yeah.
Can I make you a panelist so we can actually see you and you will be able to share your screen?
Sure.
Okay. Promote to panelist.
Can you see my screen?
Yeah, we can.
Oh, you can see it. Okay. Cool. Mine's a very quick project. It's plotting brain surfaces in Matplotlib. So what we're talking about primarily here is like cortical mesh surfaces where you have vertices and triangles, and ways to visualize data on them. So that might be cortical thickness data, or the curvature of the surface, or an fMRI activation, or a diffusion signal, or a parcellation. And the problem that I ran into about a year ago is that there's lots of nice Python-based packages for visualization, but lots of them have dependencies that are quite difficult to install, and sometimes they only work in the notebook, not for scripting. So if you want to create a large number of images, that doesn't really work. Matplotlib is almost always already installed with Python. It's a very basic package, and so I don't really see it as a dependency, but the aim was to create a Python-based visualization for brain meshes using Matplotlib only. And that's this GitHub repo that I'm proposing as a project. The basic repo now works. It can take a vector of surface-based data like cortical thickness here, and plot it on a mesh with a color map of your choosing. And it's got 1 or 2 extra features that I added that I wanted, like masking out of the medial wall. But it's still a very, very basic, simple package. And so the aim in this BrainHack would be to add some new features that are useful for people that might want to do this kind of plotting of brain surface data. So for instance, on the right, I've managed, there's a color bar, but there's no label for it. So adding a label to the color bar would be a very simple feature to add. So this is the GitHub repo. It comes with all the data we currently need. It's got a mesh file, a thickness file that you can visualize on it. And we can add some more files if we think we need to. And then, so the kind of good first issues would be, you don't need any coding experience, because one of the things that I would like is ideas for things it would be nice to see in this package. So if you have ideas for visualizing brain surface data, that'd be really helpful. A feature that I would quite like is a way to visualize flattened surfaces. So currently I have that 2 lateral medial view, but it's very limited to just that. So I'd quite like to be able to have a nice view for a flattened surface like you see on the right. I've also got an idea that I'd quite like to be able to put arrows on that surface. So those are 2 things I'll probably be working on, unless someone has some really good suggestions as well. And then the overall aim is to keep it an extremely simple, basic usage, so that you can very quickly create a brain image and without loads of extra additional dependencies. So ideal collaborators would be people who've got ideas for data visualization, people who enjoy coding visualizations, or anyone who's got experience creating testing or features that make GitHub repos more reproducible and easily installable. And so please get in contact. That's my Twitter, the Mattermost channel, or my email, or through GitHub. That's it.
Thanks, Conrad. I can already tell you that there were people that said, I really want to use that. So this means that probably they don't want to contribute to that, but they want to use it.
Well, users are great as well.
Okay, so we are done with the pitches, and now it's time to for the participants to actually meet the people that they would like to work with. So there are 2 ways that we will go through. So basically, if people want to actually interact offline per se, and basically either interact on the Mattermost channel or drop an email or things like that, it's okay. Otherwise, we can you can directly join the Zoom hacking space. That's the second link in your email, and there you will find breakout rooms. Each breakout room will have the name of each project, and hopefully in each room you will find one of the leaders of the projects. And I didn't mention this in the presentation, but I will also create a breakout room called Coffee Break, because you have seen that in the schedule there are coffee breaks. Those were supposed to be actually time away from the display. So, but if you want to just meet someone to have a chat, you can use the coffee break room. And before you leave, to take a suggestion that I just got in the chat, I will also put the link of this next Zoom meeting, that it's not this same one, here on the chat. So maybe it's easier and you don't have to go and find your previous email. So just a second, here is it. 21. Okay, so you should see the link in the chat, and with this we are done with this session. Thank you very much for coming, and I'll see you in a bit on the other side.
So, Matteo, I need to leave this open, right?
Yeah, that is what I wanted to ask you. It is
I'll
probably
join
easier
with my personal Zoom. login.
Yeah, because since I need to start that meeting, can you also stop sharing on YouTube?